Evolution, origin of life, theory of spontaneous generation or abiogenesis. Abiogenesis is a generation of the life from non-living matter. Abiogenesis, now more precisely known as spontaneous generation, this theory states that complex living organisms are generated from decaying organic substances. Organisms like mice spontaneously appear in stored grain, or maggots spontaneously appear in meat. Sir Francisco Redi, an Italian physician, was the first who disproved the theory of spontaneous generation by performing a controlled experiment. In 1668, Francisco Redi performed an experiment to check whether maggots really come from decaying meat. He did this by placing meat in a number of jars and covering some of them with fine gauze while leaving the others uncovered. Maggots developed only on the meat which are uncovered. And from this, Redi concluded that the maggots did not came from the meat but from the eggs that flies had laid on the meats. Since flies could not land on the meat in the covered jars, they could not lay eggs on that meat, and no maggots formed. Therefore, decaying meat could not produce maggots. Later, Lazzaro Spallanzani, an Italian naturalist, performed a similar experiment with broth. He put two glass flasks and sterilized them by boiling the flask containing broth. One of the flasks were opened to the air, the other flask was sealed up to keep out any organism that might be floating in the air. So microorganisms developed only in the uncovered flask. From this, Spallanzani concluded that the microorganisms did not came from the broth but were in the air that entered the flask. Spallanzani. Broth heated, cool, flask left open, wait, growth. But another one, open flask, like this. Sterilized broth in open flask developed microbes, whereas the sealed flask microbes not developed. Unfortunately, many scientists were not convinced by his experiment. Louis Pasteur, a French chemist who fully disproved the theory of spontaneous generation in the mid-80s, he performed the same type of experiment as Spallanzani. Louis Pasteur, however, allowed air to enter into the flask of sterile broth. He performed the experiment with two flasks, one with straight neck and other with a shaped neck. Flask with a straight neck allowed both air and microorganisms to enter, whereas the other flask with a shaped neck allowed only air to enter but not microorganisms. The broth in the straight neck flask became contaminated with microorganisms, but the broth in the flask with a shaped neck did not become contaminated. Therefore, Louis Pasteur showed that even though air could get in the flask, the broth did not produce microorganisms. A pictorial depiction of this. No microbial growth occurs in flask with a S-shaped neck, whereas microbes growth occur in flask with broken stem. Scientists finally were convinced that living things, no matter how small, do not come from non-living things. The present theory of where living things come from is called biogenesis. So, this theory states that living things come only from other living things. For example, maize come only from maize, and microorganisms such as bacteria can only come from other bacteria. Since spontaneous generation was now proved incorrect, many scientists begin to wonder how life started on Earth. The Earth formed about 4.5 billion years ago. It is very difficult to determine life's origin because it began at least 3.5 billion years ago. But the preparation of life had to be begun a long time before, some 4.3 billion years ago. Million of years after, under the influence of internal terrestrial heat and solar radiation, the first molecule capable of reproducing themselves and favor catalytic reactions emerged. This crucial event could have occurred 3.8 billion years ago, but in which condition was this prebiotic soup cooked? AI Oparin attempted to answer this question. Next is about chemical evolution. The spontaneous generation debate heated up again in 1924 when Russian biochemist A.I. Oparin proposed that life had arisen from simple molecules on the lifeless earth under much different atmospheric conditions than exists today. However, instead of life arising suddenly, a previous spontaneous generation theory is proposed. Oparin believed that it occurred over a very long period of time. Oparin and Haldane, working independently, proposed a theoretical model for chemical evolution. The Oparin and Haldane model suggested that under the strong reducing conditions theorized to have been present in the atmosphere of the early Earth between 4 to 3.5 billion years ago, inorganic molecules would spontaneously form organic molecules, simple sugars and amino acids. In 1929, English biologist J.B.S. Haldane published a paper in which he proposed that ultraviolet light acting on a primitive reducing atmosphere containing water, ammonia, and methane produces oceans with the consistency of a hot dilute soup containing the dilute building blocks of life. Haldane theory is most famous for its reference to the hot dilute soup of the early Earth. In 1953, Stanley Miller, along with his graduate advisor Harold Urey, tested this hypothesis by constructing an apparatus that simulated the parent Haldane early Earth. Urey-Miller experiment or Miller-Urey experiment, a classic experiment, fundamentally established that Earth's primitive atmosphere was capable of producing the building blocks of life from inorganic materials. It established that the conditions that existed in Earth's primitive atmosphere were sufficient to produce organic molecules like amino acids. Tungsten electrode, L-liter flask, condenser, apparatus for stimulating the synthesis of organic compounds on the prebiotic Earth. A mixture of gases thought to resemble the primitive Earth's reducing atmosphere is subjected to an electric discharge to stimulate the efforts of lighting, while the water in the flask is reflects so that the newly formed compounds dissolve in the water and accumulate in the flask. In 1953, University of Chicago researchers Stanley L. Miller and Harold C. Urey set up an experiment investigation into the molecular origins of life. Their innovative experiment design consists of the introduction of the molecules thought to exist in every Earth's primitive atmosphere, early Earth's primitive atmosphere, into a closed chamber. Methane, hydrogen, ammonia gases were introduced into a moist environment above a water containing flask. To stimulate primitive lighting discharges, Miller supplied the system with electrical current. After a few days, Miller observed that the flask containing organic compounds and that of some of these compounds were the amino acids that serve as an essential building block of protein. Using chromatological analysis, Miller continued his experiments, observations, and confirmed the formation of amino acids glycine, alanine, aspartic acid, glutamic acid, and beta alanine, hydroxy acids, and other organic compounds like urea, lactic acid, then glycolic acid, succinic acid, propionic acid. Thus, the Miller-Urey experiment demonstrated how some biological molecules such as simple amino acids could have arisen abiotically, that is, thought non-biological process under conditions thought to be similar to those of the early Earth. Then condensation and polymerization. Miller and Urey demonstrated the formation of simple biomolecules, but the further chemical evolution would depend on the polymerization or condensation of this monomer into polymer, formation of macromolecules like polypeptides, polysaccharides. For example, nucleic acid bases can be synthesized under supposed prebiotic conditions. In particular, adenine is formed by the condensation of HCN, a plentiful compound in the prebiotic atmosphere, in a reaction catalyzed by NH3. The other bases have been synthesized in similar reactions involving HCN and H2O. Sugars have been synthesized by the polymerization of formaldehyde CH2O in reactions catalyzed by divalent cations, alumina or clays. It is probably no accident that these compounds are the basic components of biological molecules. They were apparently the most common organic substances in prebiotic times. Proteinoids, thermal proteins. In the 1950s, Sidney Fox and co-workers, University of Miami, produced synthetic polymers by heating a dry mixture of amino acids at 150 to 180 degrees Celsius. Some show peptide-like products with molecular weights between 4,000 and 10,000 Daltons. These products bear remarkable protein-like properties with respect to enzyme activity, color test reactions, hormonal activity, and so on. Then, origin of organized structure. If complex molecules are produced from simpler ones, how might they have become organized into cells? Macromolecules aggregate into membrane enclosed droplets, conservates, and spheres as a result of intermolecular forces which exhibit some features of living systems such as organization, selective permeability, and energy, etc. Coacervates. Coacervates, term introduced by Oparin, is an aggregation of colloidal particles in liquid phase. An aggregation of colloidal particles in liquid phase that persists for a period of time as suspended membrane droplets. Formation of coacervates occur when dispersed colloidal particles separate spontaneously out of solution into droplets because of special condition of acidity, temperature, and so on. Formation of coacervates occur when dispersed colloidal particles separate spontaneously out of solution into droplets because of special condition of acidity, temperature, and so on. Coacervates show a number of interesting properties like able to exchange substances with environment, can increase in size, selectively concentrate compounds within them, and microspheres, molecular aggregates of proteinoids. Fox showed that microscopic membrane-bound spheres, termed microspheres, formed when proteinoids are boiled in water and allowed to cool. The microspheres are uniform in size, stable, and double membrane bound that appear somewhat cell like and can undergo fission and budding. Some cell like properties, such as osmosis, growth in cells, and selective absorption of chemicals, have been ascribed to them. And the spontaneous self assembly of microorganisms into coacervates and microspheres indicate that the occurrence of similar structure under primitive conditions would probably give rise to more organized structure, protocells. Later, protocells that could reproduce would be acted on by natural selection, which would act as a creative force, successively enhancing the probability of advantageous molecules, reaction, and structure. Then, membranes defined in the first cell. One of the crucial events leading to the formation of the first cell must have been the development of an outer membrane. The need for containment is easily fulfilled by a class of molecule that has the simple physiochemical property of being amphipathic, that is consisting of one part that is hydrophobic, water insoluble, and other part that is hydrophilic, water soluble. When such molecules are placed in water, they aggregate, arranging their hydrophobic portions as much in contact with one another as possible, and their hydrophilic portions in contact with water. Amphipathic molecules of appropriate shape spontaneously aggregate to form bilayers, creating small closed vesicles whose aqueous contents are isolated from the external medium. At present day, all present day cells are surrounded by a plasma membrane consisting of amphipathic molecule, mainly phospholipid. Presumably, the first membrane-bound cell were formed by spontaneous assembly of phospholipid molecules from the prebiotic soup, enclosing a self-replicating mixture of RNA and other molecules. It is not the case clear at what point in the evolution of biological catalysts and protein synthesis this first occurred. In any case, once RNA molecules were sealed within a closed membrane, they could begin to evolve in earnest as carriers of genetic instructions. They could be selected not merely on the basis of their own structure, but also according to their effect on other molecules on the same compartment. So gases on the of the primitive Earth atmosphere: CONH3, CH4, HCN, H2O, heat or other forms of energy plus water. So heat of other forms of energy plus water, amino acids, heat, anhydrous conditions, proteinoids, water, protocells, mild heat, replication of protocells, energy storage retrieval mechanism or ATP, then photosynthetic mechanism, internal protein synthesis, nucleic acid synthesis, photoribosomes, genetic coding mechanism, contemporary cells. Outline of the major steps postulated for the origin of modern cells. Once again, gases of the primitive earth, amino acids, proteinoids, protocells, replication of protocells, energy storage retrieval mechanism, photosynthetic mechanism, internal protein synthesis, nucleic acid synthesis, then photoribosomes, genetic coding mechanism, contemporary cells. Then current models. There are no two standard model for the life of Earth, but most currently accepted models built in one way or another upon a number of discoveries about the origin of molecules and cellular components for life, which are listed in a rough order of postulated emergence. Prebiotic conditions result in the creation of a certain basic small molecules or monomers of life, such as amino acids, demonstrated in the Urey-Miller experiment by Stanley L. Miller and Harold C. Urey. Phospholipids can spontaneously form lipid bilayer, a basic component of the cell membrane. The polymerization of nucleotides into random RNA molecules might have resulted in self-replicating ribosomes. RNA world hypothesis. Selection pressures for catalytic efficiency and diversity result in ribozymes, which catalyze peptidyl transfer, hence formation of small proteins, since oligopeptide complex with RNA to form better catalysts. Thus, the first ribosome is born and protein synthesis becomes more prevalent. Proteins outcompete ribozymes in catalytic ability and therefore become the most dominant biopolymer. Nucleic acids are restricted to predominantly genomic use. Then evolution of the cell. The earliest cell probably arose about 3.5 billion years ago, billion years ago, in the rich mixture of organic compounds and the primordial soup of the prebiotic times. They were almost certainly chemoheterotrophs. Primitive heterotrophs gradually acquired the capability to derive energy from certain compounds in their environment and to use that energy to synthesize more and more of their own precursor molecules, thereby becoming less dependent on outside sources of the molecule, less extremely heterotrophic. A very significant evolutionary event was the development of photosynthetic ability to fix CO2 into more complex organic compounds. The original electron hydrogen donor for this photosynthetic organisms were probably H2S, yielding electrical sulfur as the byproduct, but at some point cells developed the enzymatic capacity to use H2O as the electron donor in photosynthetic reactions, producing O2. The cyanobacteria are the modern descendants of these early photosynthetic O2 producers. One important landmark along this evolutionary road occurs when there was a transition from small cells with relatively simple internal structures, uh, the true so-called prokaryotic cells, which include the various types of bacteria, to flourishing of larger and rapidly more complex eukaryotic cells, which are found in higher animals and plants. The fossil records show that earliest eukaryotic cells evolved about 1.5 billion years ago. So, details of the evolutionary path from prokaryotes to eukaryotes cannot be deduced from the fossil record alone, but morphological and biochemical comparisons of modern organisms have suggested a reasonable sequence of events consistent with the fossil evidence. Three major changes must have occurred as prokaryotes gave rise to eukaryotes. First, as cells acquired more DNA, mechanisms evolved to fold it compactly into discrete complexes with specific proteins and to divide it equally between daughter cells at cell division. These DNA protein complexes are called chromosomes because easily especially compatible at the time of cell division. Second, as cells become larger and intracellular membranes organelles developed, eukaryotic cells have a nucleus which contains most of the cell's DNA enclosed by the double layer of membrane. The DNA is thereby kept in a compartment separate from the rest of the contents of the cell, the cytoplasm, where most of the cell's metabolic reactions occur. Finally, primitive eukaryotic cells, which were incapable of photosynthesis or aerobic metabolism, pooled their assets with those of aerobic bacteria or photosynthetic bacteria to form symbiotic associations that become permanent. Some aerobic bacteria evolved into the mitochondria of modern eukaryotes, and some photosynthetic cyanobacteria become the chloroplasts of modern plant cells. Next, theory of evolution. What is evolution? Biological evolution, simply the descent with modification. Evolution is technically defined as the gradual process in which something changes into a different and usually more complex or better form, as it is most famously used. Evolution is the process by which an organism becomes more sophisticated over time and in response to its environment. Evolution helps us to understand the history of life. Then misconception: evolution is the theory about the origin of life. Evolutionary theory deals mainly with how life changes after its origin. Science does try to investigate how life started, whether or not it happened near a deep sea vent with organic molecules came first, etc. But these considerations are not the central focus of evolutionary theory. Regardless of how life started, afterward is branched and diversified. The most studies of evolution are focused on those processes, not on origin. First, Lamarckism. Jean-Baptiste de Lamarck, French biologist, published the first truly mechanistic theory of evolution in his book Philosophie Zoologique. I'm not sure about the pronunciation. Called the theory of use and disuse or inheritance of acquired characteristics. According to this theory, the ecological conditions in which an organism lives causes it to have certain needs. These needs are met by modification of old organs or by production of new rudimentary organs. By continued use of the rudimentary structures, they increase in size and functional capacity, and by disuse, they may degenerate and become lost. Character change brought by the environment during the life of an individual become hereditary and thus can be transmitted to the next generation. Example of the evolutionary concept of acquired characteristics driven by an inner need. A short-necked ancestor stretches to feed on leaves high in a tree. The use of the neck changes the animal's heredity accordingly. The modified heredity of the ancestor is manifested by slightly longer necks in its offspring. After many generations of such behavior, descendants all have much longer necks. Lamarck would say that cave fish have often blind because their ancestors had lived for several generations in dark conditions, and in each generation the eyes had progressively degenerated through lack of use. The long neck of modern giraffe could also be explained by the Lamarckian theory. During times when food was scarce, the short-necked ancestors needed to stretch their necks to get leaves higher up in the trees. In this way, they stretched their necks. In each generation, the exercising of the neck is in this fashion caused an increase or increment in the length of the neck, which becomes genetically fixed. Lamarck's theory may sound possible, but it is unsupported by any unequivocal scientific evidence. Darwinism. Charles Robert Darwin, 12 February 1908 to 19 April 1982, was an English naturalist who proposed and provided evidence for the scientific theory that all species have evolved over time from one or a few common ancestors through the process of natural selection. This theory became widely accepted by the scientific community in 1930s and now forms the basis of modern evolutionary theory. Darwin compiled evidences for evolution by natural selection for about 20 years. Between 1842 and 1844, he wrote a 230-page essay summarizing his theory and the evidences for it. In the 1850s, he began working on a detailed multi-volume book to present his theory to the scientific community. Darwin might never have completed the book if another British scientist, Alfred Russel Wallace, in 1823 to 1913, had not came up with the same idea in 1858. Wallace formulated his theory and wrote it in an essay, which he sent to Darwin. Darwin's fellow scientists persuaded him to let them present his theory and Wallace's essay jointly at a scientific meeting. The presentation excited very little attention, according to the modest Darwin. However, the publication of Darwin's book, The Origin of Species, in 1859 changed biology forever. Darwin clearly and logically presented the idea that natural selection is the mechanism of evolution. In Darwin's own lifetime, many scientists became convinced that evolution occurs by means of natural selection. Today, this theory is the unifying one of all biology. Darwin's theory and evolution by natural selection. First, Darwin stated that variations exist among individuals of a species. Second, he stated that scarcity of resources in a population would lead to competition between individuals of the same species because all use the same limited resources. That is struggle of existence. Such competition would lead to the death of some individuals while others would survive. From this reasoning, Darwin concluded that individuals having advantageous variations are more likely to survive and reproduce than those without the advantageous variations. Survival of the fittest. Here we could see a logical argument for natural selection. First, we will be reading factors 1, 2, 3 and then some inferences, then fact 4 and 5 inferences, then last inference, final inference. Fact 1. All species have such great potential fertility that their population size would increase exponentially if all individuals born would again reproduce successfully. Next, except for minor annual fluctuations and occasional major fluctuations, populations normally display stability. The natural resources are limited. In a scale environment, they remain relatively constant. Then, first inference. Since more individuals are produced than can be supported by the available resource, but population size remains stable, it means that there must be a fierce struggle for existence among individuals for a population, resulting in the survival of often a very small part of the progeny of each generation. Then, no two individuals are exactly the same. Rather, every population displays enormous variability. Much, much of this variation is heritable. Next, inference. Survival in the struggle for existence is not random but depends in part on the hereditary constitution of surviving individuals. This unequal survival constitutes the process of natural selection. And over the generations, this process of natural selection will lead to a continuing gradual change of populations, that is evolution, and to the production of new species. Then Darwin coined the term natural selection to describe the process by which organisms with favorable variations survived and reproduced at a higher rate. An inherited variation that increase an organism's chance of survival in a particular environment is called an adaptation. Over many generations, an adaptation could spread throughout the entire species. In this way, according to Darwin, evolution by natural selection would occur. Example of Darwinian concept at evolution by natural selection. Heritable variations in neck length existed in the ancestral population, relatively short average neck length. During times of food scarcity, the longer necked individuals are able to exploit food resources that are unavailable to the shorter necked individuals, tend to survive and perpetuate their kind in much greater. So after many generations of the selective process, the descendant population has a much longer average neck length. So earlier we learned about Lamarckism. Okay, so the same example we are using to interpret the Darwinian theory. Then Darwin's theory invariably had a combination of some of the following 5 theories in mind. Evolution as such. This theory states that the world is not constant or recently created nor perpetually cycling, but rather is steadily changing, and that organisms are transformed in time. Then common descent. This theory considers that every group of organisms descended from a common ancestor, and that all groups of organisms, including animals, plants, and microorganisms, ultimately go back to a single origin of life on Earth. Then multiplication of species. This theory explains the origin of enormous organic diversity. It postulates that species multiply either by splitting into daughter species or by budding, that is, by the establishment of geographically isolated founder populations that evolve into new species. Then gradualism. According to this theory, evolutionary change takes place through the gradual change of populations and not by the sudden saltational production of new individuals that represent a new type. Then non-selection. According to this theory, evolutionary change comes about through the abundant production of genetic variation in every generation. The relatively few individuals who survive owing to a particularly well adapted combination of inheritable characters give rise to the next generation. And then next, Neo-Darwinism. Darwin proposed his theory of evolution by natural selection without knowledge of the sources of variation in populations. After mental slough were rediscovered, and genetic variation was shown to be generated by mutation, Darwinism and mentalism were used as framework of what come to be called the synthetic theory of evolution or Neo-Darwinism. According to this theory, although mutation is recognized as the ultimate source of genetic variation, natural selection is given the dominant role in shaping the evolutionary genetic makeup of populations and in the process of gene substitution. Then evidence for evolution categories of evidences supporting evolution. Evidences supporting the evolution model comes from multiple distinct areas of biology and geology. These pieces of evidences can be grouped into four categories like direct observation of evolutionary change, homology and development, then vestigial traits, then fossil record and biogeography. So direct observation in the sense small scale evolution can be observed in nature or generated experimentally in the laboratory. The classic story of a bird moth in Britain during the Industrial Revolution is an example of microevolution. Grant and Grant have recorded evolutionary change in beak shape within and among populations of Darwin finches over periods as short as 2 years. We can also drive genetic change in laboratory populations of Drosophila and other model organisms in laboratory. By selecting certain phenotypic characters, we can generate directional change in characteristics such as abdominal bristle cut number, lifespan, and avoidance to certain chemicals. Artificial selection experiments are also the foundation of agricultural improvement over the past 10,000 years. Homology and development, which we will be learning with the help of next video. Thank you.